Most biology explanations start with molecules, pathways, or structures. They quietly assume that a cell is just a container where chemistry happens. That intuition feels reasonable. And it's also the reason so much biology feels fragmented and confusing later. In this lecture, I want to replace that intuition with a more accurate one, not by adding details, but by changing the starting point. Let's start with the model most of us carry, whether we realize it or not. The intuitive model goes like this. A cell is a container. Molecules float around inside. Reactions happen because the right molecules are present. If you add the right enzymes, the right substrates, and the right conditions, chemistry takes care of itself. This model works just long enough to get you started, and then it quietly fails. The problem is that chemistry is not stable. Reactions interfere with each other. Reactive byproducts damage nearby molecules. Errors accumulate. Left alone, even perfectly reasonable chemistry drifts toward disorder. If a cell were just a bag of molecules, it would not last very long. So, instead of asking what reactions happen inside cells, we need to ask a more basic question. What problem does a cell actually have to solve? Every living cell faces this same fundamental problem. It must maintain internal order while remaining open to its environment. Closed systems die. They run out of energy and materials. Open systems destabilize. They accumulate damage and lose control. Cells must do both at the same time. This is not a minor constraint. This is the defining constraint of cellular life. Openness is required, but it's dangerous. Energy enters, but so do disruptive molecules. Waste accumulates, damage spreads. Every exchange with the environment is a risk. Cells are not protected from chemistry. They are immersed in it. Maintaining order is not a one-time achievement. It requires constant energy input, continuous regulation, active correction of error. Cells are not stable by default. They are stable only because they are constantly working to prevent collapse. And even then, failure is inevitable. The question is not whether cells experience failure, it's how long they can delay it. Once you see the problem clearly, the definition of a cell changes. Cell is not a container. A cell is a constrained system, actively managed and constantly at risk. It survives not because chemistry behaves, but because the system continuously prevents chemistry from destroying itself. This reframing explains why regulation is everywhere in biology, why growth is dangerous, and why failure is not surprising. This is the core idea to take with you. Cells do not exist because chemistry works. They exist because systems work hard to keep chemistry under control. Once you see cells this way, everything else in biology starts to make more sense. We can have the right model of a cell. We can finally start asking the right questions about regulation, growth, damage, and failure. That's where we're going next.